Hello, my dear students. Today's class is about the addition of precursors and metabolic regulators to the media. In the previous class, what we mentioned, it is about media formulation, and different type of sources which is present in the media, and the mineral requirements, carbon requirement, nitrogen sources, everything we discussed in the earlier class. Okay. Today's class is about the addition of precursors and other metabolic regulators to the media. Okay, so this is what not commonly required for the microorganism to grow, but some additional requirements we are adding to improve the product formation in a better scale. That's what it is mentioning. In the fermentation industry, some of the component of fermentation media which helps to regulate the production of the product rather than the support of growth of microorganisms. As I said, such additives included precursors, inhibitors, and inducers. Okay, all of which may be used to manipulate the progress of fermentation. So let's move to the topic. First is about precursors. Okay, so it's a name. It is saying it will the precursors which we are adding to the media, which will improve the yield of the metabolites. Okay, some chemicals when added to certain fermentation or directly incorporated into the desired product. Probably the earliest example is the improving of penicillin yield. A range of different side chains can be incorporated into the penicillin molecule. By adding the precursors, we can add the different side chains into the penicillin molecule for the better yield of penicillin. The significance of this different side chain was noted when we are adding the corn steep liquor, which will increase the yield of penicillin from 20 to 100 units. So 20 to 100 units penicillin yield is improved when we are adding corn steep liquor as a precursor. Okay, so this corn steep liquor was found to contain phenylethylamine, which was preferentially incorporated into the penicillin molecule, which will give leads to the production of benzyl penicillin, that is penicillin G. Having established that the activity of penicillin, which is laid in the side chain, it became standard practice to nowadays. It became a standard practice to add a side chain precursor to the medium, and particularly phenyl acetic acid. Okay, so it is nowadays it is very common by incorporating the side chain to make the product better yield. Okay. So researchers also showed that addition of phenyl acetic acid and its derivatives to the medium were capable of both increasing penicillin production threefold and to directing biosynthesis toward increasing the proportion of benzyl penicillin from 0 percentage to 93 percentage at the expense of other penicillin. So when compared with the other expense, it will induce 0 to 93 percentage, promote the penicillin production when we are adding phenyl acetic acid as a precursor. Okay, so phenyl acetic acid is still the most widely used precursor in the penicillin production. As I said, we can use the precursor to produce the metabolite in a large scale. For that, we are using certain amount of precursor. One of the example it is quoted here for the benzene or benzene production. We will use the phenyl acetic acid, and here it is given the certain list of precursors which commonly we are using in the fermentation industry. For example, phenyl acetic acid as a precursor for the penicillin G production, then chloride for chlorotetracycline in streptomyces. Like that, there are number of precursor it is given here. So any type of precursor we can use it for the product production. Okay. The second category is inhibitors. That's the name. It is saying that inhibitors. It will inhibit something and it will promote. It will inhibit the production of one product and it will promote the production of another product. That is called as an inhibitors. When certain inhibitors are added to fermentation, more of the specific product may be produced. More of the specific product can be produced when we are adding the certain inhibitors. So. One of the earliest example is the microbial production of glycerol. In the case of glycerol production, it is depend on modifying the ethanol fermentation by removing the acetic acid. The pathway of the glycerol production and ethanol fermentation is Same, by removing the acetic acid from the ethanol production, we can convert this ethanol fermentation to a glycerol production. That's what it is mentioning here. So for that, the addition of sodium bisulfate to the broth, which leads to the formation of acetaldehyde bisulfate. Okay, when we are adding the sodium bisulfate, this acetaldehyde is converted into acetaldehyde bisulfate. So since this acetaldehyde is no longer available for reoxidation with NADH2, it places hydrogen acceptor is taken by their place of hydrogen acceptor is taken by dihydroacetone phosphate, which was produced during the glycolysis. Okay, so the product of this reaction is glycerol 3 phosphate. Instead of acetaldehyde, it is converted into glycerol 3 phosphate, and finally it leads to glycerol. Okay, so the conversion of ethanol is changed to glycerol. That's what we can achieve. In most cases, the inhibitor is effective in increasing the yield of desired product and reducing the yield of undesirable related products. Okay, a number of studies have been made with the potential chlorination inhibitors, for example bromide, to minimize chlorotetracycline production during tetracycline fermentation. Then inhibitors have also been used to affect cell wall structure and increase permeability for the release of metabolic. The best example is the use of penicillin as a surfactant in glutamic acid production. So not only we can use inhibitors to inhibit the production of one product into another product. Here it is suggested that we can use the inhibitor to release and to increase the permeability of the metabolite. For that we can use penicillin in the glutamic acid production. Okay, so that is about the inhibitors. And here it is mentioned about the list of inhibitors which we commonly use it. For example, glycerol, we can use it for in the production of glycerol. We can use inhibitor sodium bisulfate. Okay, it will affect the acetaldehyde production, and it leads to glycerol production in yeast cells. Okay, then tetracycline production we can use bromide. Then for glutamic acid production we can use penicillin. For citric acid we can use some alkali metals. Okay, so these are the list of inhibitors we commonly use it in the fermentation industry. The final one is about inducers. Okay, so what is inducer? As the name suggested, that it will induce something. Okay, it will induce the production of some of the product. Okay, the majority of enzyme which are in an industrial interest are inducible. Induced enzymes are synthesized only in response to the presence of environment of an inducer. Okay, when we are using a particular inducer, when we are inducing in an environment, certain enzymes will be induced. Okay, so the inducer are often substrates such as starch or dextrin for amylase production, then maltose for pullurinase production, and for pectin for pectinase production. Okay, so most inducer included in microbial enzyme production are substrate or substrate analog. Obviously some of the major inducers which commonly used in the fermentation industry are substrate or substrate analog, but certain intermediates and product may also be used as an inducer. For example, maltodextrin, which induces amylase and fatty acid, and which induce amylase and fatty acid also induce lipase. Okay, and then it is now possible to produce a number of heterologous protein in yeast, fungi, and bacteria. So this includes protein of viral, human, animal, plant, and microbial origin. So it is possible to induce different heterologous protein in yeast cells and in fungi. Then, however, the heterologous protein may sometime produce some toxicity to the host and have a major influence on the stability of the heterologous protein expression. Okay, as well it is restricting the cell growth as biomass. The toxicity will be produced selective condition for surgeon cell which no longer synthesize the protein at such high level. When the production of heterologous protein, it may also lead to produce some toxicity and will stop the expression of the protein. In such cases, we need to care with an optimum growth condition, maybe achieved by not synthesizing a heterologous protein continuously and only indicating it after the host culture have grown up in the vessel, which is producing in a sufficient biomass. Okay, so therefore based on an optimum growth, we need to remove the particular heterologous protein. After it is induced, we need to remove the heterologous protein from the media. That's what it is mentioned. Inducer should have to induce or trigger the particular product, but it should not present in throughout the fermentation media. That should be removed from the fermentation. That's what it is mentioning about an inducer. Okay, so some of the example of inducer which commonly used in fermentation industry. Amylase is an enzyme, and for the production of amylase enzyme we will use starch as an inducer. Okay, then for polonase enzyme production we'll use maltose as an inducer. Like the number of inducer which was given here. Okay, so today's topic we mentioned about the precursors, the precursor inducers and the inhibitor which we are using, inhibitors then inducers that commonly used for the fermentation industry for the production of secondary metabolite, which was not commonly used, but in the certain case, if you want to improve the product production, if I want to improve the product production in an industry, we need to use such type of precursors, inducer, or inhibitors. Okay, so that's all for today. I hope you understand. So if you have any queries, you can contact me. See you in the next class. Okay, thank you.